Welcome all to this exciting new Gen webinar entitled High Throughput Proteomics During a Pandemic. This webinar was made possible through sponsorship from Bruker. I'll be your moderator for today's presentations. I'm Jeff Bugaliskas, the technical editor for Genetic Engineering and Biotechnology News, which is in its 40th anniversary year, covering all aspects of the life sciences industry. Yet this past year has been unlike any other in Gen's history, but our commitment to bringing you cutting-edge life science research has remained unaltered. And today, we're happy to bring you two presenters that are approaching one of the more difficult but extremely important aspects of the coronavirus pandemic, proteomics. In recent years, technological advances have aided clinical research efforts by allowing investigators to analyze clinical samples using high-throughput proteomic workflows, creating opportunities to analyze large numbers of patient samples, a scenario that is urgently needed during this pandemic. Additionally, proteomics scientists now have the opportunity to analyze individual patient tissue proteomes in 3 dimensions, helping us to bring precision medicine one step closer to a standard part of diagnostic reality. Now it's my pleasure to introduce our speakers for today's webinar, who are going to tell us so much more about their proteomic research and experiences. Dr. Roman Fischer is an associate professor and senior group leader in clinical proteomics at the University of Oxford, where he leads the Discovery Proteomics facility at the Target Discovery Institute. Currently, Roman's laboratory focuses on the development of high-throughput and spatial proteomics methods and their application to large clinical cohorts. And today, we're going to hear how all of that can be applied to COVID-19 samples. Then we'll turn the presentation over to Dr. Catherine Wong, who is the director at the Center for Precision Medicine Multi-omics Research within the Peking University. Catherine has been dedicated to the development of cutting-edge mass spectrometry-based proteomics technologies and the applications in basic biology and biomedical research. Recently, Catherine's laboratory developed an absolute quantitative proteomics method for mass spec, or TIM-LAQMS, which led to the discovery of new functions for the TCR-CD3 complex and its effect on CAR T-cell therapy. Today, Catherine is going to discuss how she is using quantitative proteomics to study the suppression of the human immune system during the early stages of COVID-19. But before we begin the presentations, I want to remind the audience, as always, to stick around for the Q&A session that's going to occur right after the last talk. Don't be shy. You can send in any question you want for our panelists at any time during the presentations. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. And we're going to try to get to as many of your questions as possible. All right, with that all out of the way, I'm going to turn the floor over to our first presenter. Roman, over to you.
Hello everyone, my name is Roman Fischer. I work in the Target Discovery Institute at the University of Oxford, and I'm sending this to you here as a pre-recorded talk from the confines of my kitchen. But I want to talk mostly about high-throughput proteomics and what people can do during a pandemic to help with, helping with this quite severe disease. But first I want to start with asking the question, why would you need to do high-throughput proteomics? And Ishnai Levin wrote a couple of years ago a really nice review in highlighting basically the required N number for proteomics experiments based on some modeling. And I think this is quite insightful. If we want to look at, for example, samples which don't vary between them a lot, right? This is something you would expect in tissue culture-based experiment. So if we assume we have a variation of 20% between protein abundances between samples, and if we look into this kind of modeling here to achieve a statistical power of 0.8, right, and to be able to determine a 1.5-fold change, we already would need 6 biological replicates. Now, if we want to be a bit more generous and say we only look at fold changes above 2-fold change, then we come quite close to what usually is being used in these kind of basic science experiments, a replicate of 4. And then we still have 0.8 statistical power. Now, if we have a larger variation, for example, in animal-based experiments, obviously we need higher N numbers. So a 1.5-fold change, to be able to detect that with statistical power of 0.8, we would now suddenly need 8 biological replicates. So this is something that really rarely is used in modern day proteomics, right? So there's a number of studies where you find these relatively high number of biological replicates is quite low. And this becomes even worse if we wanted to do clinical proteomics. Now here, to maintain statistical power of 0.8, and basically, let's say we have 100% overall variation. This is not unusual in a clinical sample, like a plasma sample, for example. Then to maintain 1.5-fold change at the threshold, we actually would need something between 60 and 70 biological replicates, clinical samples, blood samples per group. Now, obviously, if we only want to compare disease versus control, that still is kind of feasible. But the moment you add covariances like age and sex and so on, suddenly the number of groups that you can imagine within your clinical cohort goes up quite a bit. And then suddenly, let's assume you have just 8 of these groupings. Suddenly you look at hundreds of samples you need to analyze by proteomics. And this is really rarely being done at the moment. Now, in this review by Philipp Geyer and Matthias Mann, this was really highlighted as something that the proteomics field as a whole is going to at the moment. We want to do high throughput proteomics, and we want to address clinical cohorts of significant size with hundreds or thousands of samples. And we want to have a discovery cohort of equal size as the validation cohort. And then only then we can actually factor in things like disease severities or covariates like age and sex, disease severities, time-specific changes, but also very case-specific protein changes, or things that simply don't change at all. And we want to detect this on hundreds of thousands of proteins at the same time in a very simple experiment. Only then we can actually do correlation analysis and then hopefully be able to at some point allow us to stratify patients in disease groups that would then allow us to diagnose or prognose patient outcome or disease outcome. And this would then ultimately lead us to be able to stratify unknowns into these disease groups. So this was limited by sort of the old bottlenecks until very recently. So here we had to deal with loads of samples, and one limiting factor here was manual sample preparation, right? So if you imagine you need to digest and prepare hundreds of samples by hand, this is quite cumbersome. But also we had issues with robustness of the liquid chromatography workflow in front of the mass spectrometer. Mostly people are using nanoflow in proteomics. So this basically is very finicky and hard to maintain over a long period of time, especially on very nasty samples by nature, very dirty samples that are usually plasma samples or serum or blood-derived samples. And then we have issues with the robustness of the mass spectrometer, not so much really with stability of mass detection, but also mostly of contamination. So very often your quadrupole, for example, would get dirty quite quickly when you run plasma-derived or blood-derived samples. So this is an issue as well. This is sort of one of the older bottlenecks. Now, in the last, let's say, 2 or 3 years, machines have been developed to address these points, right? We can now these days do robot-based liquid handling and do the sample preparation on hundreds of samples within a day. We can now, using higher flow liquid chromatography, we can run, again, loads of samples, hundreds of samples per day without compromising really much sensitivity, especially considering the more sensitive mass spectrometers these days. And also the robustness of the mass spectrometer itself has been improved by dealing with sort of pre-filters towards the ion path, and then basically allowing us to run a lot of samples, dirty samples, without compromising the detection of ions in the sample. But now we face new bottlenecks, and these bottlenecks have been addressed in other omics technologies, metabolomics, for example, for a long time. But for like a modern proteomics lab, these are quite new challenges. And these already go back to the manual sample acquisition, basically the putting the sample into a consistent sort of vessel across hundreds or thousands of samples. We need to track these samples. That brings in labeling. We need to deliver the samples into a format that we can actually use in the liquid handling platform. And this is all a new bottleneck that proteomics has not been faced with sort of for a long time. And then we have to deal with primary data analysis. And with that, I mean, not really the systematic analysis, integration with other omics, and so on. There are workflows for this already, and these can be sort of modified to accommodate proteomics data as well. No, what I mean is the simple identification of peptides, and then the resolving of protein inference, right, protein grouping. And this is sort of the very basic things that we have been using for a long time on small sample sets. However, this is changing once you reach a certain number of samples and a certain number of spectra. Now, here's one of our first example in the lab. So like 2 years ago now almost, we acquired a timsTOF Pro and an EvoSep1 LC system. And after sort of working out what these machines can do, for a couple of months, we thought, okay, let's go and jump into a really big study already. Right? We specifically designed and bought this machine for high throughput proteomics. And there we go. We had like 2,600 samples from a sepsis cohort, clinical sepsis cohort, that we wanted to analyze. And this is basically a third of the samples that you see here in 10 different boxes. And you can immediately see the challenge here now. So now we have different types of labeling with different types of vessels where the samples are in. We will have different volumes of sample in these tubes and so on. So to get these into a format that you can actually use on a liquid handling robot or even for manual sample processing already takes weeks for a single person to sort out. So this is one of the bigger challenges that we have been facing. Now imagine this times 3, and then we end up ultimately with samples on Evo tips in this case. And here basically you see like 30 boxes that would accommodate these 2,600 samples. Now this wasn't everything. So we had quite a lot of additional samples that we needed to analyze. So all of these samples are non-depleted. So we don't have any capability or capacity to deplete 2,600 samples of the most abundant proteins. So they had to go in there undepleted. It's also a cost factor. But we added a number of 1,700 library samples and quality control runs. So that's meaning we have generated pools of all 2,600 samples. We made a single pool that we were injecting every 20 runs. And then we had to sort of boost the number of identified proteins. We depleted this pool using top 64 depletion, top 12, top 14 depletion. And then on top, we pre-fractionated these depleted samples just really to boost the numbers that we can detect in these samples. And hopefully then using match between runs, again, to boost the numbers of proteins that we can actually see in each of the samples. So overall, we had 4,300 mass spec runs, and using 100 runs a day is quite easy to calculate that this required like 43 days or a bit more to actually acquire the data. So we used up 3 columns in the whole experiment. Again, that was quite pleasing to see, even though it sounds a lot if you consider the time it took. But again, it's quite a lot of injections per column that the system had to do. And let's not forget, again, these are non-depleted plasma samples. So as bad as possible, really, for an LC-MS workflow. During the whole experiment, we had to clean the ion transfer tube of the timsTOF Pro only once. So that was also really pleasing to see that the machine is actually rock solid with injecting so many so dirty samples without compromising the performance. And we actually had a PM scheduled after this acquisition, and the engineer came back and said the machine is basically spotless, as new. So that was very good. Overall, we acquired in this data set 250 million PARSEF spectra, PARSEF MS2 spectra. And if you do the math here, you can see that actually the data acquisition, real life data acquisition, is 85 Hertz. And this is now including blanks and quality control ones, but also the pre-fractionated samples that are not so busy. So that means basically we had 85 Hertz plus in the actual samples for data acquisition. So out of this, we could identify 40 million spectra. So we got 40 million PSMs at 1% FDR. That sounds a bit low if you compare this with tissue lysates, for example, but keep in mind that this is plasma. So here it's quite common not to identify so many or so high percentages of your acquired data. Overall, this resulted in 5 terabytes of data. So this is quite large. Again, this is another problem, data storage. But just to upload this, if you want to process this kind of data using a large cluster like the Amazon cloud, it already takes 7 days just to upload this data to the cloud, right? Then using software called Peaks, and here we had early access to a beta version of Peaks Online, it took 2 days to import the data into Peaks. Right, this is actually quite fast if you think about the amount of data here. And then it only took 7 days for the database search. Again, this is really quite fast. And overall, this means that we can process the data faster than we can acquire them, which is a key component of doing high throughput proteomics. But then, this was end of basically 2019, and this is when this chap came along and sort of changed all plans or all ambitions to analyze this kind of data, because we joined the COMBAT consortium. So the COMBAT consortium is basically a large group of people in Oxford using a lot of different omics technologies to detect or to help with patient stratification in the clinic. And also we wanted to learn more, obviously, of the host response to the virus. So there are loads of omics technologies addressing transcriptome analysis, CyTOF of certain B and T cells, and including FACS analysis, CyTOF, and so on. And we were just one little line here, and this is the timsTOF doing proteomics on the plasma. Overall, we had 140 core samples, and these samples were sourced from the neighboring John Radcliffe Hospital in Oxford. And we had samples coming from different disease severity groups. So we had mild disease, we had severe, critical disease, and so on. We had a small set of longitudinal samples as well. And we had a very mild disease group or not symptom-free group in the healthcare workers here, which were not hospitalized. We included some healthy controls and also sepsis as a comparator for something that is basically related to a cytokine storm. And we had a few samples coming from an influenza group as well. So as you can imagine, all these different omics types require a lot of man hours or women hours. So here basically it's just a short list of people that have been involved from multiple different departments. And I'm really pleased to be part of this group because the breadth of data that was generated in this short amount of time, basically addressing a single disease, throwing all the might of omics analysis in Oxford to it. This was really quite impressive, and I'm really, really pleased about the outcome. So what we did here was that we analyzed these kind of sample group numbers here. So we have 20 healthy controls and so on, as you can read here yourself. And basically then manually process these samples. And this was done by an heroic effort of these people where we worked under Cat3 conditions, basically to manually prepare these samples. And that alone took us about 3 days to do, even with this relatively small number of samples. So this basically accumulated 559 mass spec runs that we had to acquire. That's including quality control samples, but also depleted samples, as I mentioned before. And then the whole data we dumped into FragPipe. So on here I can only recommend FragPipe and cannot highlight enough how really good this software is. It's really, really excellent when it comes to data analysis of high throughput data. It's really, really fast. And I salute Feng Chao and Alexei for creating this pipeline because I'm really, really impressed. And they have been excellent in helping us and in their support to get this data analyzed very quickly. So overall in our data set, we identified 702 protein groups. That sounds quite nice for like 10-minute runs. But if you look into more detail, you will see that this number drops dramatically quite quickly. So we had only 589 proteins that have been identified with more than 2 spectra, right? So using conservative filters that would sort of kill quite a few of these hits already. Now, if you look at how many proteins were actually covered by spectral counts in a sample, that's 160 protein groups. Now, how many proteins could we quantify from those? We have that basically filters out another group of proteins, so we end up with 223 that we can still quantify. And now how many proteins are on average being quantified in each sample? And this is only 83, right? So out of these 700 proteins, basically the nature of the sample, being plasma samples, sort of drops the number of quantifiable proteins in these samples quite significantly. And we were left using basically a filter in which we allow proteins to be carried through when they have been identified in more than 50% of each sample group. We ended up with 105 proteins that we quantified across the whole sample set. And this is basically the result. So here we see a correlation analysis, a plot of the Pearson correlation between each sample group, and you can see that we get really nice agreement following the color code here. So here the sort of symptom-free samples very nicely correlate with each other and not so much correlate with the more disease, with higher disease severity. And that was really quite nice to see. And it also indicates that we will find a group of proteins that are sort of specific in their regulation for the disease severity. Here in this cluster analysis and the heat map, we can see that actually the samples cluster very nicely according to the group. Here on the left, we have the healthy volunteers, or called healthy controls, followed by the symptom-free COVID disease. And then as we go to the right, we see increase in disease severity as the samples cluster together. And with a little bit of a block here, we have the sepsis samples, again, quite tightly and clustering together, which is nice to see. So I could now show you some of these proteins. These are just a few box plots. The main take-home message here is that we will have proteins that are very specific for disease and also are very specific for disease severity, starting with SAA1 and SAA2, but also CRP, which are all kind of part of the acute response in the host to this virus. So this kind of profiles sort of match really well with what has already been published in the public domain. So for example, Markus Walser's paper I can highlight here that has already very similar data matching to ours, but I think with a lower N number. If we put this all into a principal component analysis, we can see that the healthy controls are really tightly clustering here together and kind of form a sub-compartment of the symptom-free COVID patients, although there is some principal component 2 contribution to separate these out further from the controls. And as we go to the left, we see that the disease severity sort of increases with the sort of really mild cases here in yellow, and then towards the more severe cases in the darker colors. And the sepsis group is kind of interspersed with the disease and critical samples. So this was really nice to see that we can actually separate the disease groups even just with the 105 proteins that we could detect in most of the samples. We can then use machine learning, and this is being done here in Perseus, just also to see if we can actually predict the disease severity group based on the proteins that we could identify. And running Perseus here, you can see that along the diagonal, we can see basically the true predictions. So here we have the really mild disease, for example, symptom-free. We have 128 cases that have been predicted to be this group, and that is actually true as well. And there are only a very small number of cases where the prediction was wrong based on our data. So it was really interesting to see that we could actually also use the proteomics data to improve the classification of patients. So for example, here we have the line for the critical patients according to the WHO criteria, and 16 of those have been correctly classified. But we also find that 13 in the critical group actually are better placed in the severe group. And the other way around, we have 19 patients that are classified by our data as by the WHO criteria as severe, which would, according to our data, better fit into the critical condition. It was also very pleasing to see that almost all sepsis patients could be predicted correctly by machine learning. And this is at the end basically just a slide to show that given all the different omics types that have been used to analyze these samples, our data, the proteomics data, the mass spec-based proteomics data, actually is sufficient to allow disease classification of these patients. And this, I must say, I'm really pleased to say that this basically, the mass spec data alone would be sufficient and is actually better than sort of any two combinations or any combination of two different omics types in this consortium. And with that, I'm at an end and I'm happy to take questions.
Roman, amazing presentation and a bunch of wonderful sets of data there. Thanks for sharing that with the audience. We really appreciate that. Real quick, I want to remind the audience to stick around for the Q&A session that will begin right after Catherine's presentation coming up in a minute. Don't be shy. Again, send in any question you might have. It's really easy. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit submit. Okay, Catherine, the floor is now yours.
Good morning. Thanks Brooker and GN to organize this seminar. It's my pleasure to share some of my work of COVID-19 disease. The title of my talk today is Immune Suppression in the Early Stage of COVID-19 Disease. The experimental design is like this. We got the patient samples in about March last year. It's also really hard for us to get the sample from the hospital. So after some efforts, we were able to collect total 37 patients. We have 14 COVID-19 disease patients, and we also have 13 non-COVID-19 with other lung infection disease, and we have 10 healthy people. We use their urine sample to do these studies. We use our timsTOF Pro with their DIA passive software to analyze all those urine sample data set. After that, we have data quality control framework and pathway analysis. So the whole work has been published on the Nature Communication in November last year. After we collect those samples from 2 to 3 hospitals, we just do the normal sample precipitation and within about 200 microliter urine, and also we will use those internal retention time standard to spike into the samples. So all the urine sample from COVID-19 patients, they have been deactivated to make sure that the viral is not in the urine samples. And we then build a spectral library, and we put all the samples together and separate them into 31 fractions. For our LC condition, we use nanoelute with 90 minutes gradient, and also the column condition is we use about 75 micron ID with 25 centimeters long with those 1.5 micrometer C18 beads. To all the column has been prepared in our lab. And for the mass spec method, we use this timsTOF Pro and with the standard DIA passive software. Every 4 samples, we spike 1 sample as a QC. The data analysis, we use Spectral Pulsar software to do the DIA data analysis. So usually, when we talk about the urine proteome, the protein ID is about average from 1,000 to 2,000. And so this time, we were able to reach a very high deep coverage of the urine proteome. We quantify about 3,500 protein groups per individual urine samples, and with a very high quality of the quantitation. And so that means if you want to reach the deep sequencing of urine or any other sample proteomes, the data quality matters the most. So I'm very happy that the Bruker instrument will make us to reach this type of data quality level. So after our paper has been online, there is a third party. They download our data, they extract our data online and do the data quality evaluation with 9 features. The first one is they check our spectral library. So here shows that the distribution of the parent ions, and about 96% of the parent ions will cover 400 to 1200 m/c. So all the DIA will be performed within this window. And the second factor they have evaluated is the liquid chromatogram. So this picture shows the MS1 TIC chromatogram. We can see that we have a very good consistent continuous TIC profile, and the half peak width and the key capacity, these are 2 key factors that will affect your LC conditions. So our half peak width is about 6 to 7 seconds, and the peak capacity is very high, it's about 500. So that will make our more than 80 minutes, they are effective gradient within our 90 minutes gradient total. So they give us the excellent performance of the LC. And then they will check on our DIA window distribution. So this is shows our DIA window distribution, and we eliminate all the singly charged peaks that will interfere the other MS2 DIA window. So the peptides were collected effectively by the windows of 25 m/z. This is actually the standard window when we use in our experiment. But now I heard that they have the variable DIA window, which is, I think, they will contribute even more better for the data analysis. And they also compare our eye mobility correlation. From this very fine line, we can see that almost it's an identical eye mobility between the theoretical and the empirical values. They give us a very high stability of the teams. And this 4 diagram shows the mass deviation of the MS1 and the MS2. So the average the mass deviation of both MS, they are less than 3.5 ppm, also with a very high stability. So the instrument performance is very stable and also keep the accuracy all the time during our data set run. So then another feature is extraction eye window of the peak width and the stability. So the better chromatographic stability, the smaller this XIC window between the corresponding DIA peak and the peak extracted from the spectral library. So from this diagram, we can see that our median the extraction window width is about 3 minutes. So this also shows that our data have a very high quality. And another very important feature of those quantitative accuracy is called data point per peaks, the DPPP. So from this is the two diagram shows the data point per peak. And this DPPP factor, they negatively correlate with those chromatographic peak width and the number of DIA window. They also positively correlate with the MS spectrum scanning speed, which means you have to get a very good balance between these 3 parameters, the peak width, the DIA windows, and also the scanning speed. And usually it's about 6 to 8 dPBs will achieve the most accurate quant result for both MS1 and MS2. So we spent quite a lot of time to optimize this, and our final the DPP of MS1 and MS2 is 7. So it gives us a very high accuracy of the data. And the normalization also shows that the signal is very consistent. And we calculate so with the 1 QC sample insert into every 4 sample, the coefficient of variations is less than 19%. This is very good for the true clinical sample. Less than the 20% CV is very. So to summarize all the features I just showed you before, and they led us to reach a very deep sequencing of the urine proteome. As I mentioned, we can quantify 3,500 protein groups with very high quality of the quantitation accuracy. So we start to analyze the data set as usual. And so we firstly compare to see the COVID-19 patient together with the healthy control. And we can get the specific 1,841 protein group, which with fold change greater than 2, and the p-value is less than 0.05. And also we compare this data set with those non-COVID lung cancer disease together with the healthy control. Then we found they have a 691 protein group overlap. Within that, there are 145 proteins. They also fall to this criteria. So add up together, we obtain 1,986 proteins groups. They have greater than 2 fold change and significant p-value less than 0.05. So we will do all the functional analysis according to this amount of the protein. So from these two diagrams, it shows this is the RT-based IDs group by condition, and this is the M over Z-based IDs group by condition. We can see that the green and the the green and the red, they represent the healthy group control and also the non-COVID-19 lung disease control. And all the COVID-19, we found they have a less ID compared to the other group. And also this is a heat map. From this clear group differentiation according to the similarity, we also find that this COVID-19 group has drastically protein. They are reducing in the expression level. And in fact, we have observed, we identify 10 times more proteins. They are decreasing and in the COVID-19 patients rather than they increase the protein. So when we analyze their KEGG pathway, we can see that those reduce the protein, those have a big variation proteins, they have a strong impact on the pathways. They related to immune system and the tight junction and massive metabolics. And we also find more than 10 pathways were significantly changed. This is the Murphy's pattern. So from all those 1,986 protein, we were able to divide them into 4 types of protein pattern from those healthy control, non-COVID-19 lung disease, and also the COVID-19 patients. So the highlight in red, they are, for example, the Fc gamma related mediated phagocytosis and PPAR signaling pathway, and the NOTLA receptor and the tight junction and metabolic pathways. They all involve these 4 patterns of the protein. And this shows that the immune response of the cells. So all of the protein, basically, they are in green, which means they are reducing in the expression level. Only 2 of them, they are increasing. So this shows very clear suppression of the immune response. When we do the functional analysis and interaction diagram, we can see that this shows the panel A shows all the immune-related pathway and the protein interaction, and this is the tight junction. The majority of the protein, they are actually in green. So the major significant protein, they are the pathway, they are chemokine signaling pathway and the Fc gamma related mediated phagocytosis and the natural killer cellular cell-mediated cytotoxicities, and the antigen process and the presentations. And we have only the beta-2M and the CD48, these two proteins, they are a little bit increased in the level. And so this definitely demonstrates us that at the early stage, the humoral immunity, the cellular immunity, and the permeability of the epithelial barrier, they are drastically. And from all the proteins, we were able to pick up, we select 6 of those proteins. They are significantly different compared to the healthy control group and the non-COVID group, non-COVID lung disease group. They have 3 proteins. They are increased, which is beta-2 microglobulin, metallothionein 1G, and fluoride receptor beta. So these 3 proteins, they are significantly increased compared to the other. Another three, as I said, significantly decreased compared to the other. They are the CAM-dependent protein kinase catalytic subunit alpha, and also the apolipoprotein A4, and the lipoprotein lipase. So these 6 we pick up as our biomarker for the potential develop as the clinical diagnosis for the COVID-19 disease. And at the same time, we separate the COVID-19 cohort into 2 conditions. One is the moderate condition and the severe disease condition. We have about 9 cases of moderate and 5 cases of the severe stage. And we can see they have a very obvious difference compared these 2 groups. Even though the patient number the size is small, but we still can see the change. They are actually quite significant. And so at the moderate, the antigen processing and presentation pathway related protein at the moderate stage, they are decreasing. And then when we get to the severe patients, they actually have those increasing protein, including those PSMD family group protein, and also the T cell activation. So in the moderate patients group, the T cell activation, it has a less significant. Also the protein related, they are decreased. And when they back to the severe patient group, the T cell activation become more significant, and the molecule, the protein related to that, such as the CSK and the GRB2 and ITGB2, they actually increase at the protein expression level. So some others, they you can see from, for example, the complement activation and the cellular response to chemokine and others, they started to also getting activated. So this data shows that at a later stage, for example, when the disease migrate from moderate to severe type, the immune responses, they are starting to get activated. So they basically get boosted when the disease become more severe. So this is another picture of the heat map to represent these moderate cases and the severe COVID cases. You can see that they have a clear separation, and from some of the pathway or the protein, they clearly to be activated at this level. And also we were able to select 2 proteins as a prediction marker, or they at least to reflect this migration from the moderate to severe stage. The one is the immunoglobulin lambda variable 3 to 25. So this molecule, you can see from the moderate to severe, they have, and compared to the healthy control, they are actually increased trend, and with a very high significant, the p-value is equal to 0.004. And another molecule is elongation factor 1 alpha 1 and the EF1A1, and it shows a decreasing trend from the healthy people all the way to moderate COVID people and the severe patients. Based on the previous data, we proposed a 2-stage model for COVID-19 disease. The patient infected by the virus, when they are at the moderate or light stage, the mainly the response of the body is immune suppression and the very severe impairment of tight junction and massive metabolic destroyed. And when they progress to the second stage, then it will cause the organs damage, and finally will be the cytokine storm. So here are our conclusions of this little project. The first is an ultra-deep urine proteome of COVID-19 was achieved by an advanced mass spectrometry technology. We used the Bruker TIMS Pro TOF instrument with their DIA passive software, which is also called by 4D DIA, and we are able to obtain a very high quality data. And also we provide an important molecular basis for understanding the clinical symptoms of patients with proposed a two-stage model. And we were able to find out some of the characteristic markers which have very high potential to be validated to develop as a diagnosis marker for detection of COVID-19 disease. And in fact, we are doing that using the clinical cases and mouse model. And the last, the immune suppression at the early stage of this disease is very important observation, because that would suggest us we should pay very good attention on the dysregulation and the clinical treatment at the beginning. So for example, if the monoclonal antibody drugs targeting on the interleukin-6 is it really suitable for treatment for treating patients at the moderate or the light stage, that may be a question. Finally, I would like to thank Professor George Fu Gao. He's the director of China CDC, and he gave us a very good support and help us to get the patient samples in different hospitals. And also thanks my group, Dr. Yang Chen, Gordon Gao, Weiming Tan, Nan Zhang, and Serena Wong. So they work together on this project. And also the funding from the Outstanding Technology Talent Program and the Research Fund of the Central University. And the most important, thank you for your attention.
Thanks, Catherine. Really wonderful presentation and some amazing research there that we hope will have a tremendous impact on this disease. To the audience, the Q&A is going to begin in just a few moments. I just want to remind you once again to submit questions for our panelists. All you need to do is click the Ask a Question tab on the right-hand side of your screen, type in your question, and hit Submit. Remember, no question is too small. Before we start the Q&A, though, I want to introduce everyone to our guest Q&A moderator today, Dr. Gary Krupa, who is the Vice President of Proteomics at Bruker. Gary has over 30 years of experience in the life sciences industry and is leading expert in the field of mass spectrometry. So we welcome him for his efforts today. Welcome, Gary. To the audience, thanks for sending in those questions. We're going to get to the Q&A, so just bear with us for a moment as we transition into the Q&A session.